Assalamu alaikum dear students, I hope you all are good. Uh, Today we are going to study pyruvate oxidation. Yesterday we have read about the glycolysis, and you already know that the glycolysis takes place in the cytosol, and we read that how glucose is broken down by a set of reactions or a series of reactions into two molecules of pyruvate. Fine, this is pyruvate, this is a 3 carbon compound. Fine, so how glucose is broken down into two molecules of pyruvate through a set of reactions is called glycolysis. That's also called as Embedon-Meyerhof pathway, and the net gain of ATP during glycolysis is 2 molecules of ATP and 2 NADH. Fine, so I hope that is clear to you. Now the pyruvate generated in glycolysis, it has to be oxidized. It has to be oxidized to enter the Krebs cycle. Now this pyruvate lies in the cytosol as glycolysis takes place in the cytosol. Fine, so let me tell you, now this pyruvate is a charged molecule and it is transported by a protein carrier to the mitochondria of the cells, as the Krebs cycle occurs in the matrix of the mitochondria. So here I have taken a small picture of the mitochondria. So fine, so this pyruvate is transported to the matrix of the mitochondria, but only in eukaryotes. In prokaryotes, it remains in the cytosol only, and there the Krebs cycle takes place in those. But here in eukaryotes, it is transported into the matrix of the mitochondria. Here what happens? Oxidation reaction takes place in the presence of enzyme coenzyme A. This is coenzyme A. And what happens? During this process, 2 NADH are formed. This NAD positive, it is reduced and NADH is formed, and carbon dioxide, 2 molecules of carbon dioxide are released. You can see here molecules of carbon dioxide are released, and what happens here? This acetyl coenzyme A is formed when the pyruvate plus coenzyme A, they add up and they form the acetyl coenzyme A. Now this acetyl coenzyme A enters the Krebs cycle. This enters the, not the pyruvate enters the Krebs cycle, but what enters the Krebs cycle? Acetyl coenzyme A, and this reaction is known as pyruvate oxidation. Here the pyruvate is oxidized. Here the pyruvate is oxidized and carbon dioxide is released and NADH is formed. 2 molecules of NADH are formed and 2 molecules of carbon dioxide are released. So in eukaryotes, this reaction takes place in the matrix of the mitochondria. Fine. Here you can see a carboxyl group is removed from the pyruvate. Here this carboxyl group, which is highlighted, C double bond O, it is CO2, it's CO2, and this is called carboxyl group. It's removed from the pyruvate and released as carbon dioxide. Now what happens? NAD positive is reduced to NADH, here you can see, and an acetyl group is transferred. This fine, acetyl group is transferred to coenzyme A, and the acetyl group is transferred to the coenzyme A, resulting in the acetyl coenzyme A. And as a result, what happens? We get this acetyl group. It is transferred to this. It's added to the pyruvate. This group is removed, and as a result, we get the acetyl coenzyme A. So let me tell you, it is a junction between glycolysis and Krebs cycle. You cannot say pyruvate oxidation occurs in glycolysis. It doesn't occur in the Krebs cycle. But what it is a junction between glycolysis and Krebs cycle. I hope I am clear here. So next is, in eukaryotes, it must enter the mitochondria with the help of a transport protein. In eukaryotes, it enters from cytosol into the mitochondrial matrix with the help of a transport protein. Fine. And the reaction takes place in the presence of an enzyme, and that enzyme is pyruvate dehydrogenase complex. Please remember this. This reaction takes place in the presence of an enzyme called pyruvate dehydrogenase complex. So we are done with the pyruvate oxidation, and finally what we got? We got acetyl coenzyme A, which can enter the Krebs cycle. Fine. So here we have the citric acid cycle or the Krebs cycle. So please have a look. Here we have pyruvate. Let me start. The pyruvate we got from the glycolysis, and in the presence of an enzyme, pyruvate dehydrogenase, what it gets converted to? It gets converted to acetyl coenzyme A. Now this acetyl coenzyme A, along with oxaloacetate, this oxalo-OAA is a 4-carbon compound. Here you can see 1, 2, 3 and 4. It has 4 carbons, and it combines with acetyl coenzyme A, and it forms citrate or citric acid as the first carbon compound. Here you can see citrate or the citric acid is the first carbon compound here, and hence it's called as citric acid cycle, because the first carbon compound formed is an organic acid, and that is citric acid. Fine. And this reaction takes place in the presence of an enzyme called citrate synthase, and water is consumed in this reaction. Fine. And now what happens next? In the presence of an enzyme aconitase, this citrate gets changed to isocitrate. Fine. Its configuration changes and it gets changed to isocitrate. See here, this is isocitrate. Now what happens? This isocitrate, in presence of an enzyme isocitrate dehydrogenase, fine, it in presence of the enzyme isocitrate dehydrogenase, it gets dehydrogenated, and as a result what is formed? Alpha ketoglutarate, and in this process NAD positive is reduced to NADH. Fine, we get NADH here. It's reduced and we get NADH here, and carbon dioxide is released in this process. Fine. So we are already 2 molecules, but I have this cycle here, it's a 1 turn of the cycle, but we are taking into account two molecules. So here 2 NADH are formed and 2 molecules of carbon dioxide are released. Fine. So for the sake of understanding, only 1 molecule has been taken, but we take into account generally we have 2 pyruvates liberated from this glycolysis, so 2 acetyl coenzymes enzyme A's, and hence here we have 2 NADH formed and 2 carbon dioxide molecules released. And finally what we get by this reaction? We get alpha ketoglutarate. And now what happens to this alpha ketoglutarate? It in the presence of an enzyme alpha ketoglutarate dehydrogenase, further it gets dehydrogenated and NAD positive is dehydrogenated, and NADH are formed and carbon dioxide is released. And we get succinyl coenzyme A. This is succinyl coenzyme A here. And now what happens to this succinyl coenzyme A? It changes to succinate. Here it changes to succinate, and what is formed here? Here GTP is formed, guanosine triphosphate. Here ATP can also be formed. In animals, here GTP is formed, guanosine triphosphate, but this guanosine triphosphate later forms 1 molecule of ATP here, and in plants directly ATP is formed, adenosine triphosphate. So here 2 molecules of guanosine triphosphate or ATP are formed, 2 molecules of ATP are formed here. Fine. And what happens to this succinate? In the presence of succinate dehydrogenase, and what happens? FAD. Fine. FAD gets changed to FADH2. FADH, it's also an energy rich molecule, and it gets reduced to FADH2, and it changes to the succinate, it changes to fumarate. Fine. And upon consuming water in the presence of fumarase, it changes to malate, malic acid or malate. These are organic acids. And finally this malate changes to oxaloacetate. In the presence of enzyme malate dehydrogenase. So this is the Kreb cycle. It takes place in the matrix of the mitochondria. Fine. So what we begin from? We begin from oxaloacetate, and this changed to citrate or citric acid, or then isocitrate, and then alpha ketoglutarate, then succinyl coenzyme A, then succinate, then fumarate, then malate, and finally we have oxaloacetate. So let's take an overview of the glycolysis and the Krebs cycle. Glucose here changed to 2 pyruvate by the process of glycolysis. I have not written here, but please take it as glycolysis is changed to 2 molecules of pyruvate, and as a result, what is the net gain? There is net gain of 2 ATP and 2 NADH. Fine. And what happens then? These two pyruvate, they undergo oxidation reaction, and as a result, 2 molecules of carbon dioxide and 2 molecules of NADH are released in the process. And this acetyl coenzyme A, when it enters the Krebs cycle, what happens? Upon combining with oxaloacetic acid, it liberates, you can say, 4 molecules of carbon dioxide plus 2 molecules of ATP or GTP here. Later that GTP is converted to ATP. Fine. 2 molecules of ATP plus 6 NADH and plus 2 plus FADH2. Here let me show you 1 molecule of FADH, and here 2 molecules of FADH, or yes, 2 molecules of FADH are formed. I have not written here, but it's FADH2 2 molecules. So as a result, how many ATP are formed? 4 ATP. Fine. 10 NADH and 2 FADH are formed in the glycolysis and the Krebs cycle. So I hope I am clear till now, and tomorrow we'll be starting with the oxidative phosphorylation, and we'll be also studying the anaerobic reaction, in which we'll be studying the fermentation. So please recapitulate the things, please go through glycolysis and Krebs cycle, and please let me know if there is anything I can help with. Thank you so much. Stay blessed, stay safe.